Good morning, everyone. It's my pleasure to be here with you today, and I would like to thank the SSBMT board for the invitation and Kite for the sponsorship. Today I'm going to present our local experience at the Ministry of National Guard Healthcare in Riyadh for the Chimeric Antigen Receptor T-cell therapy for B-cell malignancies. So as we know, leukemia and lymphoma in general consider one of the top malignancies within our population, and it has a huge burden on patient overall. And we know that relapse ALL and as well B-cell ALL and as well as relapse large B-cell lymphoma have dismal outcome and very limited options. Over the past few years, there has been an interesting way of therapy, which is the use of chimeric antigen receptor T-cell therapy for management of specifically CD19 B-cell leukemia and lymphoma. And the first publication, which was for relapse refractory B-ALL, showed impressive results, which led to the first approval of such therapy. Subsequently, multiple publications showed the use of CD19 CAR T-cell using different construct and different way of manufacturing showed an interesting finding and similar overall response rate. And this to the multiple approval of such product for either large B-cell lymphoma, for relapse refractory follicular lymphoma, and relapse mantle cell lymphoma as well. As we can see here from the ZUMA-1 trial, after a median follow-up of more than 4 years, the median overall survival was around 25 months. And this is an example for one of CAR T-cell treatment targeting CD19 for relapse refractory large B-cell lymphoma. Subsequently, the investigators, they move the use of this therapy to the second line treatment for highly refractory, primary refractory large B-cell lymphoma. And for ZUMA-7, Axicel showed superiority over standard of care, which is autologous stem cell transplant, as well as Lysocell in the Transform study, which showed superiority in progression-free survival. In general, there has been a huge interest in such therapy and huge development using different targets, different immune effector cells, and this has led to the currently at least we have 6 different products for more than 12 different indications targeting CD19 and more recently BMSA for BCMA for multiple myeloma. So we thought that this is really an important type of therapy and it's been the standard of care all over the world, and we thought that this treatment must be offered for our patients, and we started to form a CAR T-cell working group. This is way before the COVID era, thinking how to bring this type of therapy and make it local for our patient. So we formed this committee. It was a joint forces between the SCT division in pediatric and adult, as well as nursing department, pharmacy. Also, we enroll other departments such as ICU, neurology, infectious disease. And we form like regular meetings weekly and sometimes twice a week with established agenda and assignment. So the aim from this working group is to identify institutional needs for cellular therapy in general and close the gap and also to identify key personnel and specialize in such therapy, and also to standardize the institutional guidelines, and also to communicate and coordinate with cellular therapy providers. We establish a patient journey pathway until the infusion, and patient for each single step within the patient journey, starting by identifying the appropriate patient, selecting the correct patient, and for the correct indication, then clearing the patient pre the apheresis, followed by what type of bridging therapy, and this followed by clearing the patient pre infusion. We develop extensive documents and guidelines just to ensure patient safety through this journey, and also to establish a quality monitoring system. As well as the most important aspect is post-infusional monitoring as well. So establish extensive documents just to make sure that we are delivering a safe therapy for our patient, and we keep them under close monitoring. We establish electronic forms to make sure that we document every single aspect of the treatment procedure. We establish order set for each phase of during the patient journey and for each specific CAR T-cell product. Also, we establish educational activity, risk mitigation, nursing education, and e-learning for the nurses and in-service sessions, as well as establishing patient educational materials. This is an example for what kind of activity during the engagement with, for example, Kite, when we engage with them to establish the CAR T-cell in our institution. So around 4 to 5 months before the infusion of the first patient, we had the kickoff meeting with the Kite team, and this followed subsequently with quality audit, and shortly the IT training happening, and followed by clinical training, and then the quality training as well, and followed by the dry run. And shortly after the dry run, we were able to have the site activated, and a few weeks later we collected our first patient. So far we have a number of cases done in our institution. We collected 9 adult patients, and also 3 pediatric patients were collected. And so far, we infused 5 patients in adult and 2 patients in pediatrics. The patient infusion adult was on first week of January this year, and subsequently followed by pediatric infusion was in the 6th of February this year. This is a summary of patient outcome that we infused. So far, as I said, we infused 7 patients, 5 adult and 2 pediatric. And you can see here the square indicating that time line showing the assessment. And we have those who we did, they were available for assessment, they were in complete remission. So our longest follow-up for our first patient is around 19 to 20 weeks from now. And we just infused 1 patient. Our last patient was infused 2 days ago. So this is just a case discussion for one of the cases that we treated. This 36-year-old male diagnosed with in the local hospital with diffuse large B-cell MNGHA, stage 4, disease with bulky left femur mass. The IPI score of 3 and CNS IPI 3. He received 6-cycle R-CHOP followed by 2 doses of high-dose methotrexate CNS prophylaxis. End of therapy assessment showed complete metabolic response. And because of the bulky mass, they consolidate with radiation with 40 degrees to the left femur. However, 3 months after the completion of R-CHOP, patient presented with the chest wall mass showed relapse DLBCL confirmed by biopsy. He received RGDV 1 dose, and he was referred immediately for CAR T-cell as refractory DLBCL based on the data emerging from the ZUMA-7 trial. So at our institution, when we evaluated him initially, we had a PET scan. Actually, he had 2 cycles of RGDV while waiting for his referral. And then when he presented to our institution, actually his chest wall still persistently avid, and he actually developed new lung lesion despite 2 cycles of RGDP and REP hyper metabolic activity, as well as basal of skull finding. And we biopsied the lung nodule, which again confirmed the disease. And we did FISH, which showed only rearrangement of BCL6 only. So lymphocyte collection happened within 2 weeks of presentation to our institution. And subsequently, 80 weeks later, CAR T cell were infused. He developed resistant CRS of grade 1. He had only 1 dose of tocilizumab, and subsequently his CRS resolved immediately. And I can show you here his disease response assessment. So this is his, on your left side, his PET scan pre-CAR T cell infusion. You can see the lung nodules and chest wall mass, which completely resolved in his day 30 assessment. So this is just an overview of what cases we had using the commercial CAR T cell. We know that the current available CAR T cell or currently approved CAR T cell has a lot of limitation, including like relapse rates, toxicity, and as well as the logistics, cost, manufacturing time delay, and so on. So there has been a lot of effort to overcome these challenges, and research is still ongoing. And if you can see, the R&D size for CAR T cell is expanding over the past few years. This is a big numbers for overall cellular therapy in general. But if you can see, the first one for CAR T cell is one of the most active area of research. And within the CAR T cell, most of the investigational effort is within the preclinical stage. But as you can see, that there is more than 500 ongoing clinical trials for CAR T cell around the world. And some of those actually clinical trials, they are in the phase 3 stage. So we thought that we should also do something, and trying to localize such important treatment. So we had a collaboration with Miltenyi to run a phase 1 study using CD19 specific construct to be manufactured locally in King Abdullah International Medical Research Center. And so we had multiple meetings with the company, and we established a study protocol that was submitted to the IRB and approved by the IRB, and we also complete investigation brochure and the IND as well. And we actually, in communication with the Saudi FDA, looking for the approval of this phase 1 study, cellular therapy. And as I want to show you here, our colleagues from the stem cell therapy. This is the advanced cellular therapy manufacturing team at Chimeric, and this is the Prodigy machine when they run actually their first validation run, taking cells from healthy volunteer. And I want to share with you that we were successfully able to produce our own CAR T cell from healthy volunteer with around 50% transduction efficiency. So in the end, I'd like to thank all the participants over the past few years to get this important therapy available for our patients. And special thanks to Ms. Rodina and Ms. Inam, who our nurse manager in stem cell division, adult and pediatric, who were really were extensively active and working hard to make sure that we had every single document done and completed, and we did make sure every single possible training has been implemented for the nursing as well as for the whole team. And also special thanks to Inam, who helped me to prepare those slides. This is a picture for our team. Of course, we need to thank everyone involved in the process, our colleague in stem cell therapy lab, Department of Nursing, apheresis lab, as well as other departments. Thank you very much, and happy to take any questions.